Today we will discuss about effect of KRAS on T cells and myeloid cells, and we will also understand whether this effect of KRAS is only uh, observed near the tumors or it can also observed at the other sites. So in order to check the effect of KRAS mutations, they developed a mice with a following doxycycline driven KRAS. That is, when the doxycycline is introduced, then the KRAS mutations are shown and the tumor progression occurs within the mice. And if the doxycycline is induction is stops or injection of doxycycline is stopped, then the KRAS mutations goes off and the tumor regressions will occur. So this is how they develop their mice models, and they check first they check the effect in two different kind of mice model. First one is this PIKP mice model and this PKT mice model. So in the PIKP mice models, what they find out is that when the KRAS mutations are off, and when the KRAS mutations are off, they observe this different kind of immune cells after 5 days of the stop or 5 days of the stop injection of this KRAS. And what they have find out is that the T cells infiltration goes up near the tumor sites. That can be seen here in the panel B, that CD3 plus T cells, CD8 plus T cells, CD4 plus T cells goes up, and also the CD4 plus FOXP3 T cells, CD4 plus FOXP3 P3 minus T cells goes up. And in terms of the myeloid cells, they can see the decrease in the myeloid cells populations such as CD11B plus, CD11B plus, LY6 negative, LY6G plus, and LY6C, CD11B plus, LY6C plus, LY6G minus. So all these three myeloid populations and its subpopulations goes down when the KRAS mutations are turned off, indicating that this KRAS mutations uh, has a role towards affecting the CD to affecting this T cell population and the myeloid cell populations. That is, when the KRAS mutations are off, the T cells population goes up, whereas when the KRAS mutations are on, the myeloid cells population goes down. And these things are quantified on the panel D. In the top figure of the panel D shows about PIKP model, where we can see that when the KRAS mutations are off, they see the increase in CD8 T populations and its sub category of the T populations, whereas a decrease in the myeloid populations, but they do not see any significant changes in the NK populations and CD19B populations. And similar results they have observed for the PIKT tumors. And the panel and this bottom side of the panel B and panel C also this shows the results about the PIKT mouse or mouse that is infected with this PIKT tumor. Here we can see that similar results are observed, that is populations immune cells related to the T cells increases, whereas immune cells related to the myeloid cells decreases. And next they check the immunostaining the populations of about the T cells and the myeloid populations. So when the KRAS mutations are not there, they do not see any immune cells infiltrating to the site of the tumors. That is, there is no CD4 plus populations and there is very less myeloid cells, that is CD11B populations. When the KRAS mutations are on, they see that increase in the myeloid populations. And when the KRAS mutations are off, even after 5 days, they see some of the myeloid populations are still present, but they see the increase in the CD4 plus cells. And after 2 weeks, they see the complete decrease in the myeloid populations and the increase in the CD4 plus populations. And these things remains to continue even after 4 weeks. Similar scenario remains to continue even after 4 weeks. That can be seen in this immunostaining, and these things are quantified in the panel F, where we can see that when it's come to the CD4 plus populations, they can see CD4 plus populations are high when the KRAS mutations are off at different time intervals, and similar results they have observed for the CD8 plus populations also. But for the myeloid cell populations, it goes down when the KRAS mutations are off. But for the after 5 days of the KRAS mutations are off, they do not see a significant decrease. But for the 2 week and 4 weeks, they see that significant decrease. And when they check the different T cells populations, when the KRAS mutations are on and off, they find out that in the case of PIKP, they can see that when the KRAS mutations are on, there is a more of the myeloid populations. 67% of the myeloid populations are present. When the KRAS mutations are off, so this panel, first figure of the panel G, shows about these KRAS mutations are on. So in the one case, on the one side, it shows that it has a high populations of these myeloid cells, and in the second side, they shows that it has this high. These are the different kind of myeloid cells that are present. That is CD11B plus Ly6 minus Ly6G, and also CD11B plus Ly6 minus Ly6G, and the other cells, that is CD19B cells, the CD11B plus Ly6 minus, and also these ungated cells. These are the non-gated cells, and the CD8 plus T cells. T cells populations are very low. This is the main things that they wanted to point out, that T cell populations are low, whereas myeloid cell populations are high. When the KRAS mutations are on, the KRAS mutations are off, they see the reductions in the myeloid cell populations and increase in the T cells populations. And similar results they have observed for the PIKT models. That is, when the KRAS mutations are on, they see the high myeloid cell populations, 79%. The T cell populations are very low, around 2%. But after 5 days of when the KRAS mutations are off, they see the reductions in the myeloid cell populations and increase in the T cell populations. That is, myeloid cell population goes down from 75 to 47, and T cell populations increased from 2% to 9% after 5 days. And thereafter, they did check the different kind of immune cells, and the effect of these KRAS mutations are off on the spleen has also been observed, but it is presented in the supplemental figures. Here they have find out that in the spleen they do not see too much difference when on the immune cells when the KRAS mutations are on and off, indicating that the effect of KRAS mutations are in the tumor immune microenvironment only, or near the tumor sites. And when they check the different kind of immune cells, apart from the different subpopulation of T cells and myeloid cells, they find out that it's a mixture of tumor regression and tumor promoting tumor regressions and tumor promoting cells are formed when the KRAS mutations are off. For example, this TIM3, it helps in the it results in the T cells exhaustion. That is, it helps in the tumor promotions are increased when the this KRAS mutation are decreased when the KRAS mutations are off. That can be seen here. That is, tumor T cells exhaustions are decreased when the KRAS mutations are off. That is, for CD4 plus T cells and CD8 plus T cells, it's non-significant. It looks like it's increased, but it's statistically non-significant. And for this TBET plus, they see the similar things, but TBET is the tumor regressions. This TBET is the tumor regressions cells and its population. Tumor regressions immune cells and its populations increases when the KRAS mutations are off. That can be seen here. That is CD4 plus T cells and CD8 plus T cells. The percentage of TBET is high. And when they check for the cytokines, that is LLFNG, its expressions also goes up, and it has tumor regressions properties. When its expression goes on, the KRAS mutations are off. And in terms of the dendritic cells, they see that different kind of dendritic cells also goes up when the KRAS mutations are off. That is, CD8 dendritic cells that has CD8 plus 86 plus markers goes up. when the KRAS mutations are off, and also for this CD40 plus markers, the T cell that has a marker of CD40 plus also goes up when the KRAS mutations are off, indicating that the tumor regressions are also expressed when the KRAS mutations are off, but the tumor suppressions factors also expressed that are shown in the supplemental figures. So I hope you understood the effect of KRAS on the T cells and myeloid cells. To some extent, it looks like confusing as this effect, even when the KRAS mutations are off, it also promotes some of the tumor promoting cells, tumor promoting factors or tumor promoting cells. So I hope you got an idea of got an basic idea about the effect of KRAS on the T cells and myeloid cells. It's basically predominant on the tumor immune microenvironment, but not on the not at the system level. That has been checked with the change in the immune cells, immune cell compositions at spleen when the KRAS mutations are on and off, and that results are shown in the supplemental figures. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.